Have you ever wondered what happens after you eat a meal full of the necessary dietary fats? How is something that isn't water-soluble digested in our bodies? My name is Alyssa, and this video will help you understand this concept by dissecting what happens to lipids as they are digested and absorbed. We'll start off by investigating just that process, the digestion and absorption of lipids. We'll then introduce the term chylomicrons, and we'll see how they are important to transporting lipids throughout the body. After watching this video, you will be able to map out the process of lipid digestion and absorption, and be able to answer questions specific to the compounds present throughout these processes. Lipids pass through the mouth and stomach undigested, and so it is not until they reach the small intestine that digestion begins. Lipids must first pass through enterocytes, and are then absorbed into lymphatic capillaries known as lacteals. However, in order to reach the lacteals, the lipids must first be digested into smaller portions. Lipids are hydrophobic molecules and therefore form large, indigestible droplets in the aqueous environment of the intestinal lumen. In order for digestion to occur, bile acids first break down these large droplets into smaller droplets. These acids are synthesized in the liver from cholesterol, secreted from the liver, and stored in the gallbladder until they are needed. Bile acids are said to be amphipathic. This means that they contain both a hydrophobic portion, in this case the green cholesterol backbone, and a hydrophilic portion, which in this case is all of the red hydroxyl groups found on the molecule. These bile acids dissolve the large lipid droplet. This occurs because the hydrophobic portion dissolves in the lipid, while the hydrophilic portion dissolves in the aqueous lumen, making the lipid droplet more soluble. This creates emulsion droplets that have a greater surface area to volume ratio, increasing the possible areas for digestion. When bile acids have finished their work for the day, they are reabsorbed in the ileum and recycled, and any undissolved lipid droplets pass through the colon and are excreted. Pancreatic lipase and colipase then further solubilize the emulsion droplets to even smaller micelles. Micelles are key components of lipid absorption, so let's see how this process works. Most ingested fat is in the form of triacylglycerides. These are big, bulky molecules that do not pass through the membrane easily because they tend to form large clusters. Pancreatic lipase and colipase are responsible for breaking down triacylglycerides so that they become smaller, more manageable molecules that can be absorbed. By cleaving the outer two fatty acids from this molecule, 2-monoacylglyceride is released, as well as 2 free fatty acids. These molecules are smaller and are more soluble than the emulsion droplets formed by the triacylglycerides, and so they form smaller micelles. Micelles are spheres of lipids with their hydrophobic fatty acid tails oriented towards the center and their polar ends oriented out towards the aqueous environment. These spheres contain monoacylglycerol, individual fatty acids, cholesterol, and the fat-soluble vitamins A, D, E, and K. Micelles spontaneously break apart and reform, which frees individual fatty acids and monoglycerides. These molecules diffuse towards the brush border of enterocytes. Here, cilia aid in lipid absorption because of the increased surface area they provide. The components of the micelle are small and nonpolar, so they can diffuse through the membrane only after the micelle breaks apart to release them. The micelles themselves are too large to diffuse into the cell. What happens to the components once they are absorbed into the cell? Let's take a closer look at the inside of enterocytes. Once the fatty acids and monoacylglycerides have diffused into the enterocyte, they enter the endoplasmic reticulum. Here, they are used to synthesize new triacylglyceride molecules, which are then packed into complexes called chylomicrons. Take note that the process of forming chylomicrons only occurs in the endoplasmic reticulum and only in the enterocytes of the small intestine. But what all goes into a chylomicron? Phospholipids and apolipoproteins form the outer layer of the complex. These molecules protect the hydrophobic components from the aqueous environment because they are amphipathic. These hydrophobic components are the triacylglycerides, cholesterol, and vitamins A, D, E, and K. The function of chylomicrons is to carry these hydrophobic components throughout the body. So let's investigate how these complexes leave the enterocytes. Chylomicrons are synthesized in the endoplasmic reticulum, packaged in the Golgi apparatus, and leave the enterocyte via exocytosis across the basolateral membrane. Because chylomicrons are relatively large, they cannot be taken up into the intestinal capillaries. Instead, they diffuse into special capillaries known as lymphatic lacteals. This completes the process of digestion and absorption of lipids. Once in the lacteal, the chylomicron will travel with lymph until emptying into the subclavian vein via the thoracic duct. Once in the bloodstream, the chylomicron can deliver triacylglycerides and cholesterol to various tissues such as adipose tissue, skeletal muscle, and the liver. Now that we understand the theory of lipid digestion and absorption, let's take a look at a question regarding monoacylglycerides in these processes. The answer is D. Remember that monoacylglycerides are a component of micelles, which help molecules diffuse into enterocytes from the intestinal lumen. There, the molecule enters the endoplasmic reticulum and is used in the synthesis of triacylglycerides to later be packed into chylomicrons. Fantastic work if you got that question correct! In this video, we examined how lipid droplets are broken down by bile acids and then pancreatic lipase and colipase to create micelles. These micelles aid in the diffusion of lipids into the cell by releasing individual fatty acid molecules so that smaller molecules can diffuse into the enterocyte. In the endoplasmic reticulum of the enterocyte, chylomicrons are packaged and released into the lymphatic lacteal, where they transport lipid-soluble components throughout the body. Now that you've watched this video, you should feel confident in your knowledge of lipid digestion and absorption. Good luck studying this topic for your MCAT.